Hello, my name is Regina Villarreal, and I will be talking about NIM's biofungicide effect on coriander microgreens. We noticed that cedarwood farms have encountered fungal contamination in some of their harvests, especially those of coriander microgreens. So we wanted to demonstrate the effects of neem extract as a natural biofungicide for these coriander microgreen cultivations. So to begin with, what is neem? Neem is a natural pesticide and fungicide that has proven to be effective in the elimination and prevention of fungal growth. Therefore, we wanted to test the hypothesis that neem extract will have an effect on the growth of contaminated fungi in coriander microgreen cultivations. For our experimental design, there were a couple of variables that we took into consideration. Our independent variables included the type and source of treatment, the concentration of the treatment, and the time of cultivation. The dependent included stem length, percent of germination, percent of contamination, and wet biomass yield. And our controlled were the substrate amount, water, light, and temperature conditions. So we wanted to see how these variables would interact with each other and the type of results that we would get about neem's biofungicide effect. To actually test our hypothesis, we followed a protocol for microgreen cultivation in which we had 4 trays, 3 treatment replicates, and 1 control tray, all with, like I mentioned, the same amount of substrate and roughly the same amount of seeds. To our 3 treatment trays, we added a neem extract, and to our control tray, we didn't. And then we let them cultivate and germinate for 15 days, taking measurements at days 7, 10, and 15 of percent germination. And at day 15, we cut all those microgreens that germinated to see if the results that we gathered at day 15 of cultivation, if they were significant. We did 2 types of ANOVA test. For the length of the stem, we used a one-way ANOVA. In our case, we found out that the treatment had no significant relationship with the stem length. And then to see if our type of treatment had some type of effect on the percent of germination, we did a factorial design ANOVA, and we tried to determine if either treatment, days, or both had some sort of effect on the percent of germination. And our results from that ANOVA were that they both do have an effect on the germination percentage. Now moving on to our protocol proposal, we obviously took into consideration what was taught by us by cedarwood farms, how they cultivated their microgreens, and we tried to replicate it. And for our in vitro protocol for the germination of microgreens, we also looked at some literature that did somewhat similar experiments. In my opinion, our in vitro germination proposal has some cutting-edge biotechnological elements. For example, tissue culture, as plant tissue culture in vitro is fundamental in this biotechnology area. And another cutting-edge biotechnology aspect that could potentially be used is genetic modification, which would help with the physical aspect of the microgreens, its nutritional content, and its resistance against diseases or fungi. We also know that the microgreen market has grown over the last couple of years and will continue to do so, especially in the areas around gastronomy or like food, restaurant, business / industry. So I believe these protocols could be of use for those people interested. And so in my opinion, our protocol does have an added value, especially in the area of sustainable production, since our protocol is designed to be the most efficient as possible, which could be a very important added value, as well as the reduction of diseases or fungi contamination, because by following our protocol, we are reducing the possibility of our microgreen cultivations getting contaminated by fungi, which goes in pairs with another added value that I believe is consistent quality. By having a well-defined protocol that you can follow every single time you do a microgreen cultivation or germination process, you can ensure that the quality among all of the trays are roughly the same. Finally, I believe that our project has many advantages that make it valuable in a biotechnological aspect. Some of these advantages would include a controlled and constant microgreen production, improving the efficiency of our germinations or cultivations, and reducing the effect of fungi contamination and sustainability by making sure that our protocol is the most efficient as it could be. However, with every advantage, there are also some disadvantages, which I believe could be a high initial cost. By using fungicide, it's obvious that the cost could increase a little bit. It's also a little bit tricky to kind of nail the concentrations and how you apply them at the first time, so it's definitely a learning process. We also have to take a lot of consideration, which could be a disadvantage per se, of the regulations and norms that are implied by the Mexican government, so we gotta make sure that we follow those. And obviously, the biggest disadvantage is that these microgreen cultivations, as they're very short cultivations time-wise, like it's only 15 days, there has to be constant monitoring of its growth and what's going on within the germination. In conclusion, neem extract had no discernible impact on stem length, but it significantly affected germination percentage. It's also important to mention that all of our trays remained uncontaminated and that there was a high cell-viability percentage, which would confirm neem's biofungicidal properties and that this extract is not harmful to the cells. Thank you.